Yeah, for this talk, like I did last time, what I wanted to give is a brief sort of description about what metabolic optical imaging is, how it evolved, and then coming down to phasor and how you use it. And the idea sort of follows some of Dave's talk with historical of fluorescence and things like that. So it's slightly historical, then slightly more, and it ends in the last paper I published. So as Lionel mentioned, I'm in Georgetown. That's the main campus. I live in the opposite side of the campus, which is the medical center, which doesn't look as nice and as cool as the old campus does. But anyway, that's our picture. So, okay, so going down to, let me go back to 1962 and start talking about the first paper that sort of started this field. And the paper was intercellular oxidation and reduction stress in vivo, by, and it's from Beaton Chance. I think it's literally the paper that started our field. And this is not imaging, this is spectroscopy. So, and we'll go from the spectroscopic aspects of it to the imaging aspects as we go along. Right? So what they're doing is they were putting these two objectives. And can you see my cursor? Just, I hope you do. Right. So
Yes, Suman.
okay, awesome. So they put one of the objectives in the kidney, the other one in the brain, and what they are looking at is how the intensity of fluorescence in the NADH changes over time as they put different gases in the mouse, right? So the idea, DPNH is actually NADH, and there's a spectral shift between the reduced and oxidized form of it. And the idea here is basically you would start with oxygen and nitrogen cycles, and you would see the intensity going higher when the nitrogen cycle, when the cycle operates, right? So that's fluorescence increases at the bottom direction. So between start and stop in kidney and brain, you can start seeing this behavior, but you can basically sort of monitor what is happening to the mice, brain and kidney, when it is going under different metabolic changes because they're inhaling more oxygen or nitrogen. So that was the original idea, right? The problem is this is single photon, which means if you try to take it to the imaging plane, it becomes very difficult because the energy of the single photon at 300 something nanometer is so high, it's not going to work in cells. So there was a delay, I would guess. And then over at Webb's lab in 2002, so after quite a big delay, they created the two-photon fluorescence spectroscopy and microscopy of NADPH and flavoprotein. So what I really like about this paper is it gives you the two-photon excitation spectra of the NADH, NADPH, FAD, and LIDH, which is taken into the solution, right? Both excitation and emission spectra. So this allows now to do two-photon, and obviously with the energy of this, each of these photons is being half of the single photon. Now you can try to think about applying this in the microscope. And with the advent of new laser and the microscope, it went from basically a spectroscopic measurement to now a imaging system. So the question obviously is like, what can you see and what can you do? So this is a simplified energy diagram taken from Kyle Quinn's paper. And the main reason I put this up is the simplified version of all the energy diagrams because there are so many steps in there. And so if you look at it, we can look at the coenzymes, which are autofluorescence, when you excite around 370 nanometer in single photon, and 400 nanometer for FAD, right? And out of these species, there are two, NADH and FAD, which are actually fluorescent. FADH2 and NAD plus are dark. So we can't see them with fluorescence. We can only see NADH and FAD. And using those two, the idea was, can we actually monitor cell metabolism using those two. The first thing people came up with is something called optical redox ratio, which is basically the intensity of the FAD divided by the total intensity, or there are different versions of this. There could be FAD by NADH only, there could be NADH by this sum, or NADH by FAD. There's different versions, so you have to be careful when you look at the old papers, how they define it, because that would matter. Now, what this showed, this optical redox ratio is correlated to NAD plus by NAD plus by NADH ratio. And this ratio is known as this is the actual redox ratio in the system. So by looking at the intensities, you can monitor what's happening inside the cells or tissues. So now, just to give a brief description of where this thing stands, you have your NADH exists in protein bound and protein and free NADH. They have similar excitation spectra, similar emission spectra, but they have a different lifetime, very different lifetime. And NADH lifetime matters what it binds to, the coenzyme it binds. It's a coenzyme, so it depends on the enzyme it binds to. Same thing with the FAD. Again, bound and free FAD have different spectral. And free NADH, I just did it twice. Anyway, doesn't matter. So first thing is to how to validate it, right? You come up with a new technique, you have to validate it in some way. And so this is the validation is the old school way of method using LCMS, the mass spectra, liquid chromatography mass spectroscopy. And if you look at the ratio of the NAD plus to NADH by NAD plus, and if you look at the optical redox ratio, they're very highly correlated. There are different types of cells in there. And that sort of tells you how well the old school measurements of metabolic changes are correlated with the optical redox ratio changes. Now, this is stem cells which are going under different types of differentiation. So MSC propagation, osteogenic and adipogenic differentiation. Now, if you look at the redox ratio, you can definitely see the difference in different parts of the cells and how they're changing in different types of differentiation. Now, what this is the experiment, the LCMS doesn't give you, and imaging does, is the spatial heterogeneity of this system, right? So that cannot be seen, and I'll come back to that. Now, there are other people who also have done biochemical analysis using the optical redox ratio. So this is in stem cell, this is in pancreas, in epithelial tissue, to show that they are highly correlated, and you can use the optical redox ratio as a proxy for LCMS. Now, again, the inter and intercellular variability that you can see in optical redox ratio, that cannot be measured using the old school liquid crystallography, right? Now, the other validation is you have to one of the most commonly used systems is seahorse assays. And you can measure the oxygen consumption rate or proton production rate. And the idea would be, can the optical redox ratio compared with OCR, right? So to do that, what you do, you take you basically change the mitochondrial metabolism. You can either use oligomycin, which would stop the ATP synthase. You can use FCCP, which basically stops this whole cycle, basically, because it uncouples the NADH electron transport. And then you can use rotenone or antimycin A, which basically blocks, rotenone blocks complex 1, antimycin A blocks complex 3. And by blocking these complexes, you change the metabolism. So, and the idea is, let's see what happens in the seahorse assay, and can we replicate it with optical redox ratio? So these are cells with baseline oligomycin, FCCP, and antimycin rotenone A. And you can see the NOCR, which is on the right. You can see the shift, and you can see the exact similar behavior on the optical redox ratio. Now, this has larger error bars because you have spatial heterogeneity built into it. Each cell is measured individually, compared to oligomycin, where each OCR, where it's a measurement of the whole plate of cells. So it's a single measurement versus there's a spatial heterogeneity and sample heterogeneity that is built into the optical redox ratio measurement. Similarly, cancer cells under normoxia or when they are given hypoxia, followed by deoxygenation, you can see what happens to the optical redox ratio. You can see what happens in OCR, and you can basically see that as the metastatic potential of these cancer cells changes, the highly metastatic potential has metastatic cancer cells have more post-hypoxia change in the redox ratio. So that was another way of defining how, especially in cancer cells, this was used. Right now, a lot of this work was originally done in breast cancer cell because the work comes from Nirmala Rajaram's lab in University of Wisconsin, and so up to this point, I told you about how optical redox ratio works, right? But then, as Lionel mentioned, we talk about FLIM and fluorescence lifetime imaging, which I guess Enrico talked about before, and Alex went through it, right? So why do you do that? One of the important part is that if you look at the optical redox ratio, it's defined by the intensity ratios of FAD and NADH. Now, the difficulty is intensity ratios are related to the quantum in their molecular fraction, right? So if you have only two species, FAD and NADH, that's fine, but that's not true because we have bound and free NADH and bound and free FAD. So each of these numbers is dependent on the fraction of this because they have such a different quantum in or lifetime, which is related to quantum in. So if you have a quantum in of 0.45 and 0.02, you can think about if you have an equal mixture of this free NADH is only going to contribute a very small fraction to that total number. So that's why it's not very sensitive. So the question then becomes, what can you do? And well, we can actually look at lifetime. So the free NADH, when the two rings are collapsed, has a very short lifetime, as shown here, 0.3 to 0.4 nanosecond. When they bind to an enzyme, these two rings go away from each other, they fall up, and the fluorescence increases as quantum yield increases, but also the lifetime increases much higher. Now, we can measure that by looking at just how long the molecule stays in the excited state. So basically on an average lifetime basis. And the big thing is independent of concentration. Now, that's great, but we now have to correlate this back to the optical redox ratio or OCR rate, right? So the first paper that started the field of flame and redox measurements is from Damien Bard. And they were looking at MCF10A human breast cancer cells using multifold fluorescence. And what they're doing, they're just looking at number of cells. So basically confluency versus the lifetime. And what's the free and protein bound NADH ratio is. And keep an eye on this ratio because that's the most important thing here, is this line here. So in a normal cell, growing under condition, this increases as your confluency increases. Now, your serum starved state, this increase is much less pronounced than here, right? This is increasing from 1 to 2.5. This is increasing from 2 to 2.5, so it's less than half. Now, when they're treated with potassium cyanide, which basically produces glycolysis, you see similar decrease in the free to bound NADH ratio as you are increasing the confluency. So it sort of starts behaving like a serum starved cell. So that was the first indication that fluorescence lifetime of NADH can be used for looking at these sort of metabolic changes. Now, one of the first paper from Melissa Scala was they are looking at basically different precancerous cell. So these are low grade, this is high, middle grade, and this is normal, low grade, and high grade. And this is going in depth. So this side is the redox ratio. This is the average NADH lifetime. So as you are going to more cancerous tissue, you are going towards a lower number in terms of the redox ratio. And as you're going deeper, you are increasing the redox ratio. The similar thing you can see here, as you're going towards more high-grade precancerous tissue, the lifetime decreases. And as you're going deeper, the lifetime decreases even more. So, and if you look at the protein contribution of protein bound, so alpha 2 here, because it's a bi-exponential feed, you can start seeing that as there's a decrease in the amount of bound NADH towards the cells. So it's becoming more and more free, more and more glycolytic, which is what is predicted by changing the metabolism to more Warburg-like metabolism. So this has been, again, done in, I kind of like this paper because they're continuously monitoring by changing topical reagent with a gravity-fed tube on top of a mice brain. And basically, they're looking at what's happening to the fluorescence intensity. And what they did, they use control two types of glycolytic inhibition, then two types of Krebs cycle inhibition, electron transport chain inhibition, oxidation phosphorylation inhibition. So by doing all of these, they can basically follow exactly what is expected. And I'm not going to go much into detail, but they can follow the difference. It changes. But, you know, the funny thing is this is 2007, but it basically goes back to similar experiment in Beaton Chance in 1962. So that's the, you know, we sort of repeat ourselves eventually by more complex methods. Now, when you compare these measurements with the OCR optical, with the Seahorse assay, and if you look at the idea here is, if you're free to bound NADH increases, which is more glycolytic, your redox ratio, the way they mention it here, would decrease, and the OCR would decrease. And if they do mitochondrial stress test, you can see by oligomycin and FCCP, the changes are correlated between the OCR and free to bound. And similarly, you can see with the glycolytic stress test is also correlated. So when there is an increase in one, there's a decrease in the other one. That's what you are expecting, right? Because these two are opposite. So whenever there's a increase in the black, you see a decrease in the red. So great. So it works, which is, and again, Chiara Stringari, who was before me, and Lionel in Indico's lab, and Kira Pate, I think, Kaida, I think her name, they actually showed the exact similar behavior with the Wnt signaling pathway in glycolysis and angiogenesis in colon cancer. The reason I didn't mention it yet because I haven't gone to Phasor yet. So these are done using Phasor. So Phasor, now coming down to Phasor and its properties, and you guys already heard about Phasors. I'm not going to go very far, but basically the idea is, when you have fluorescence intensity decays or lifetime decays shown here in these four colors, you can do a cosine and sine star transform, which is shown again in the dotted line and the black dotted and straight line. And if you could transform them using Fourier, you can transform them to the Phasor place. And because these are calculated mono-exponential decays, they will appear along this circle, which we call universal semicircle. So shorter lifetime would appear here, and as we are going in longer lifetime, it would appear here, as shown in this direction and as shown in this direction. So why do Phasor? Multiple reasons, but a couple of really important points are the reciprocity principle. And what it means is, you can have an image of a cell, you can select out, you can calculate the Phasor properties from this each of these pixel to the Phasor plot, because these are transformation happen in each point, each pixel, and color them accordingly. So now you can separate out different parts of the image. However, you can also select out part of the image and see where those Phasor points appear. The conversion between these two gives us the direct correlation, so that we can go from the image space to the Phasor space, back to the image space. So it becomes very intuitive in nature. And another thing is the linear combination principle, which is, this is shown in solution, but it is valid for a pixel, and as Alex talked about before me. So if you have two samples, which is different lifetime, if you mix them, the phasor point of the mixture would appear exactly in the line that coincides in between the original two phasor points. And the distance of this point, the purple here, from either the red or the blue, would be dependent or would be inversely proportional to the fractional intensity of that particular species. So now you can think of, we can have this and calculate back to what the fractional intensity of the each phasor is. So it's a quantitative way to calculate metabolic changes. So again, this is shown between free NADH and bound NADH. You have the shortest lifetime for free, longest for this, and these are just calculated decays with different amount of bound NADH, right, built into it. If you transform them into phasor plot, you can see the proper transformation always appear along this line. That line, we call it metabolic trajectory. And we can take the metabolic trajectory, we can calculate back, and we can see as a plot number of pixels versus fractional intensity of free NADH, what is the shift in the metabolic changes. So the more shift on this direction becomes more glycolytic. And that allows us to, again, go back and take a look at biological systems under drug or other manipulations, right? Originally from Chiara's paper, you can actually add something like potassium cyanide, which would force it to be in reducing condition, or hydrogen peroxide, which would force it to be in oxidizing condition. And you can see the shift in metabolism as shown in by the color map here in the images. You can similarly do this with high glucose or sodium glychloroacetate, where you can produce glycolysis or oxidative phosphorylation, and you see the cells moving. So we did similar experiment just to double check a few weeks ago. So these are phasor plots for multiple cells. This is the origin position. This is blown up. So this is the control, which is called TMSO1, but it's basically the control cells. As you increase oligomycin, it becomes more glycolytic. As it add to deoxyglucose, it becomes more oxidative phosphorylation. And you can see from the color map that same behavior, as I mentioned, is becoming more and more glycolytic with oligomycin. But please notice that there is a cell-to-cell variability, and there is a lot of intercellular variability when we talk about these changes. How do we do the phasor analysis? Well, obviously first thing is image acquisition, and then we do a calibration. And calibration is usually a sometimes difficult process in microscopy with FLIM. And the way phasor is easy because we can use a fluorescence dye. So let's say we measure a fluorescence dye that comes along here, but we know the lifetime of that fluorescence dye is supposed to be here. So first we do a correction for the modulation, and then we correction for the phase angle, and we force it to be in this point. Now you can use that same exact calibration on the unknown samples, and that gives you the calibrated phasor plots. You can use also scattering sample because they have basically a lifetime of 0, so you force it down the other way, bring it back, and that would give you the calibration procedure. So that gives us the calibration. So this is something that we published back in 2018 in the Nature Protocols, how to do each of these steps completely carefully. And this is basically taken from that. So once you have your calibration, you reference your sample, you can do masking, you can do image filtering, which is basically increasing the signal to noise in the phasor space without changing the actual image space. Then you can select out and basically analyze reference file based on biological fluorescence, which can be done for 2-3 components or average positions, like I showed in the last graph. You can also do freight, concentration, pH, ion based on the biosensors present, right? So that it gives us a lot of different ways to look at this system. The reasons, well, is fit-free, is visual in nature. The decays and phasor properties are assigned to molecular species and not lifetime. And that is very important to remember, because you can have something which has a multi-exponential lifetime, which is very difficult to analyze when you mix it with something else in a biological system. But phasor doesn't care. Even if it's a multi-exponential lifetime, it's a position in the phasor space. And if it's mixing with another position, it would be a linear combination. So it allows us to look at that. Now, phasor is an image. So we can use image analysis procedures. One of them is shown here. So you can do filtering. So this is 3x3 filtering or 5x5 filtering. And as you are doing filtering here, the actual image gives the same resolution as you took the original intensity image. There is the reciprocity principle, which I mentioned before, and the linear addition properties of the Phasor space, which is, again, I sort of talked about before. So one of the things from Rupsa's work, who was a post-PhD student in the lab before, she was looking at metabolic deprogramming astrocytes in Huntington disease. And again, I don't really have a lot of time to go into that, but what she saw is between wild type and Huntington disease, if you see an increase in the free NADH in the young mice, but at the old mice, you don't see it. Since striatum, which is supposed to be susceptible, you do see in young mice a large decrease in the bound NADH fraction, or in it becoming more glycolytic, which you don't see in the cerebellum, which is not supposed to be affected. So a word of caution, and this is work with Lionel. Be very careful about proper fitting and transformation when you are looking at this data. So this is showing, if you look at the historical context of phasor, you see different directions, different positions of bound NADH. And a lot of that comes from either proper transformation or not proper measurement from both perspectives. So as you know, like I mentioned, there are different lifetimes of bound NADH depending on what they are. But one of the big problem is when you take data in the time correlated single photon counting, which is the most common method in a sense, that when you do a transformation, this is a Fourier system, which means you have to have a repeating signal. So if you cut the signal, which is often done, so this black window here, so this is the repeating signal of fluorescence lifetime. If you cut it right here in the black, you are not doing a proper transformation because this is such a small window. And the decay doesn't decay to zero, so it's very hard to do this transformation. That creates uncertainty and that creates impropriety. But if you take the data like what Indigo developed, where if you take the data where there's a repeating signal, as you've shown in the red, you can actually do a proper transformation. And the thing is, the reason people don't do it is because if you took the black, you can actually work on 80 megahertz, and your window is 12.5 nanosecond. So you can get faster, more number of photons. But if you use the other way, you have to go up to 25 nanosecond, which is 2 repeating windows at least. So your time of acquisition increases by double, basically. And what happens is if you do improper transformation, you can have different trajectory, which means the images would look not right. And this is just to show that FLIMbox, which is not a TCSPC instrument, it's a modulation instrument. You can always do proper transformation, but Baker and Hickel, which is a TCSPC instrument, if you use gain of 2, which is basically this proper transformation window in red, you can actually get a same transformation as FLIMbox. But if you use gain of 5, which is only looking at up to 8 nanosecond of that window, you can actually get a very long transformation. So we have shown this by taking images and measuring solutions and things like that. So we have done that. And the other thing that I didn't put in here is when you mention bound NADH, and Lionel can tell you much better because he's the biochemist in all these experiments, but it's not easy to make 100% bound NADH. You have to force the molecule because there's a KD, there's a equilibrium constant. So you have to force it to go to that bound space. Now, what happens with fixation? So this is work from Jenu, who was a postdoc with Michelle back in the day, and this is after he moved to Wisconsin. To look at what they did, they looked at 2 types of cells, and they fixed the cells and wanted to see what happens. So now, if you look at the mean lifetime in NADH, the between the 10A and 231, the one is a triple negative base epithelial cell line, the other one is a non-cancer, one is non-cancerous, one is cancerous, and 10A is non-cancerous. So you see the decrease in lifetime. So these are more glycolytic in live cell. They're still more glycolytic in fixed cell, but in very different amount, which means you can't compare live versus fixed, but you can compare between live and fixed. So they did the same thing with sodium cyanide or 2,8-deoxyglucose, and basically you see the behavior that you expect to see, but just in a different extent. They also looked at, they plotted, this is a plot of the individual co-localization histogram where you can see same behavior in live and fixed cells. So it works, transforming into phasor plot. Again, it still works, just you can't compare between samples. And this is looking at cancerous tissue, where you can still see between the the KRAS knockout and normal pancreas, the more cancerous has a shorter average lifetime. So we're still seeing glycolytic effect as we expect to see. Just again, it's not comparing apples to oranges. You have to compare apples or you have to compare oranges, right? But that's not all. We can also look at something else. It's an oxidative stress on phasor plot. Same excitation, but sometimes when there's lipid droplets, you start seeing a separate signature. So this is Rupsa's work again. And what she was doing, she was feeding HeLa cells with oleic acid to induce fat droplets. And as she induces fat droplets, she starts seeing these secondary sort of lifetime changes, which comes from away from the metabolic free and bound NADH. And so this is the NADH map. This is what she calls long lifetime species map. And what she did then, she took the same cells and she did long lifetime maps, and she did CARS, coherent anti-Raman spectroscopy, and to look at that. What she showed is these droplets are coming from fat droplets, but not all fat droplets have this long lifetime species. She did third harmonic generation, which looks at the boundary between refractive indexes and is very good at fat droplet boundary. And you can see something like this, where there's a large fat droplet, which has long lifetime, is the same area which has a third harmonic signal. She also did Raman spectra to show that this lipid droplet, this what is present in this long lifetime species actually is different from oleic acid and actually coincides with the droplets, right? Okay, so now it's great because it can be used. That's index. So yeah. So it's also cool because you can use this as a sort of indicator. So this is normoxia and hypoxia. As you are inducing more and more hypoxia, you start seeing more and more lifetime changes. And that you can sort of see with the LLS imaging. So as you are inducing normoxia, hypoxia, you see this shift in the hypoxia with 48 hours, and this is reversible. So if you put it back into normoxia, it eventually comes back. So this is kind of a cool paper from Rupsa that allows you to look at this, this long lifetime. Now we have done a lot of quantification, and this is sort of where all this multi-component analysis started. So the idea is if you have only bound and free NADH, that would be along this line. But as you have long lifetime, it will start, depending on how much it is, it will start dragging the phasor more towards it. So you can actually sort of calculate these and just show that when you have three species, they will always appear within this window, that is within this triangle that is occupied by the free, bound, and long lifetime species. And you can actually do a projection of this map back to this metabolic trajectory and can calculate what's the fraction of free to bound NADH is. You can also calculate what's the amount of this fraction of rate. So we did that a few years ago, and just to show that how this works. So these are mice fed with normal diet or a very high fat, high cholesterol diet, which is basically American or Western diet, right? That's why it's called Western diet. Now, if you do that, this is a normal diet. You don't really see it's mostly metabolic changes. However, as you see, when you feed it with fat diet, you can start seeing this behavior. Now, we can quantify that. So we use the same system to look at a liver system if you this year, where we are basically it's a way of doing ischemia reperfusion injury. So what you do is you block it off and you so you clamp the liver, right? And then you open it up and you let it blood supply for 45 minutes and you recover it and harvest it after 24 hours and then look at under the microscope. So the idea here is basically the similar model for liver transplant surgery. So where you take the liver, you transplant to a different place and then when the blood comes back in the transplanted person, it creates injury. So it's a model for that. So if we look at what's happening in the cell, so these are on the top is non-injury, at the bottom is the injury. These are control diet. This is a control with normal diet and high-fat diet. And this is normal diet, high-fat diet, but it's a treatment with N-acetylcholine. And the most important part I wanted to show you is this. If you look at the glycolysis changes, so increasing glycolysis, when you have fat diet with injury, there's a large increase in the injury. But when you have NAC, which is a prohibitor of this injury, it decreases the injury to happen. You see increasing long lifetime species whenever there's a fat diet, but the injury or the livers don't become as glycolytic as high fat diet would allow when you have a drug in effect, basically. So it also FLIM also allows us to look at spatial heterogeneity. So you can actually map out the long lifetime. You can create a map of the free to bound NADH. You can look at this as a fraction of changes. Looking at it from heterogeneity perspective. The last thing is, what else can we use this signature for? So we can take the fluorescence, we can take the phasor, we can map out where these droplets are, and we can look at the quantification of the sizes. And that allows us to look at between, so these are different diets under different treatment, injury, non-injury here and injury there. And it allows, and this is just the average calculated from there. It allows you to look at something is very difficult is macrosteatosis or microsteatosis, what type of droplet formation is happening. So this is just an overall thing that we did in the paper, so I'm not going to go ahead. And finally, Phasor allows us now to look at 4 components, which is Balance and Alex's work. And part of that, that they looked at multiple components. They also looked at this blind resolution, which Alex talked about, to how to calculate this is, again, liver between high fat and low fat diet, and you can see how the redox ratio changes. And with that, basically,